Not that kind of cycle.
If not that kind of cell cycle, what kind of cell cycle are we actually talking about? Okay, so you can see a definition on the screen there of the cell cycle, but what does that mean? Well, new cells are needed for an organism to grow or for parts of it to grow, or to replace cells that are worn out or damaged. However, they also need the same genetic information as the original cell, and so they can do the same job. And so these three phases of interphase, mitosis, and cytokinesis are incredibly important to maintain that genetic information. Okay, so in each cell there's a nucleus which has lots of chromosomes which are tightly coiled strands of DNA, each containing many genes. What's a gene though? Well, a gene is a small packet of information that controls a characteristic or part of a characteristic of your body and is a section of a strand of DNA on one chromosome. Humans have got 46 chromosomes that are arranged in 23 homologous pairs. I'm just kind of covering pairs 4 and 5 there, but you can see this is a karyotype of a human being with 23 pairs, the 23rd being X and Y, showing that this is a male. Homologous pairs means homo means the same, and so they should be in the pair because one has come from mum and one has come from dad. Okay, so body cells have a life cycle, and GCSE asks us to split it into 3 identifiable stages. Although in reality, biologists can split this into many more stages, but we don't need to complicate matters at this stage. Stage 1 of the cell cycle is interphase. During this stage, all organelles are being increased so that the cell can get to a much larger size so that it's able to divide back down again when needed. All the organelles, including the cytoplasm and the cell membrane, are increasing in size. Remember, all of these cell organelles are highlighted in my video, Basic Cells for GCSE. So if you've forgotten any of those, take a look back at that. Also during interphase, DNA is replicating and copying itself. So by the end of this phase, there will be double the mass of DNA in the cell than normal. This is the longest stage of the cell cycle. Stage 2 is mitosis. This is actually split into 4 phases. Prophase, where the DNA coils up and chromosomes become visible in like an X-like structure. Don't confuse this with the X chromosome and the sex chromosomes because it's the shape of all the chromosomes here when they've replicated. Spindle fibers form and the nucleus dissolves in prophase. The next stage is metaphase. That's where the spindle fibers that are formed, these protein strands, grab hold of the centromere of the chromosome and they line the chromosomes up along the center of the cell, which we call the equator, like the equator of the planet. The next stage within mitosis is anaphase. This is where these spindle fibers that have got hold of the centromere contract and they pull the arms of the X-shaped chromosomes apart. These arms are called chromatids. It drags them to the ends of the cell. These are the poles, if we carry on using the planet analogy, and that's what we call them in biology. And then we go into telophase, where the nucleus forms around the newly assembled chromatids, and you can see 2 nucleus now in the cell as it starts to divide into 2 new cells. Stage 3 is cytokinesis, and this is where the cytoplasm and the cell membrane divide once and for all, and they produce 2 genetically identical daughter cells at the end of this process. Each one of these genetically identical daughter cells then goes out on their very own cell cycle and enters interphase. Let's take a look at this life cycle again in slightly more detail. So, stage 1, interphase. You've got a normal body cell there. We've only showing 2 homologous pairs just to make it a little bit easier so we can see it clearly. During interphase, everything copies and duplicates itself, as does the DNA. Here we're looking at a chromosome, but it's now got 2 arms. It's made of 2 chromatids joined in the middle by a centromere. At this stage, the cell has double the mass of DNA that it ordinarily would have. Here we can see a selection of chromosomes like the ones we saw earlier in homologous pairs, but in their duplicated form so that you can see that they're all in that X shape. So all of the organelles, including the DNA, have replicated in this stage. Moving on from interphase, we go into the first stage of mitosis, which is prophase. In this stage, all of the DNA coils up so that you can really see those chromosomes, as we just saw on the last screen in those X shapes. Before that, it was just very fine strands of DNA. Next stage is metaphase, and that's where these spindle fibers, as you can see by the black lines, have grabbed hold of these chromosomes by the centromere and line them up down the equator of the cell. And as they move into anaphase, these chromatids are pulled apart to the poles of the cell. And then we go on to telophase, where you can see that the chromatids are collected at the opposite poles of the cell, and a nucleus will form around those, ready for cytokinesis, which is stage 3, whereby the cytoplasm and the cell membrane divide completely, and we've got 2 genetically identical daughter cells. And each one of those will go on to start its own interphase. Time for Mr. T's top tip. Well, there's 2 today. Number 1, use the phrase genetically identical daughter cells to describe the end result of mitosis and indeed the cell cycle. And number 2, state that both the cell membrane and the cytoplasm divide during cytokinesis. You have to name both of the organelles in order to show your deeper level of understanding of the process. Right, let's have some application of cell knowledge. So here's that diagram again. It's quite crowded, but hopefully you understand it now. And the question is, if a cell entering interphase has 6 picograms of DNA, then what is the mass of DNA when the cell is ready to leave interphase? And you can work out what the mass is going to be at the end of the interphase by understanding that all the DNA is replicated during interphase. So at the end, there will be double the amount of DNA, and so there will be 12 picograms of DNA there. It doesn't matter what unit they say, as long as you put that unit afterwards, you should know that there will be double the amount at the end of interphase. Then the next part of the question is, how many chromosomes are there at this stage? Well, even though there is double the amount of DNA there are still the same number of chromosomes. There's 46 arranged in 23 pairs. However, this time they are arranged as 2 chromatids joined at a centromere. Okay, so don't forget that mitosis is for growth and repair, and that a child grows due to interphase. That's when the cells are getting bigger, everything's replicating, and it takes up more space. That's what growth's about. More cells, and each cell taking up more space. It's as simple as that. Some organisms use mitosis to reproduce asexually, producing complete clones of themselves. But most don't. Most produce sexually. And so use meiosis for sexual reproduction. Please don't confuse meiosis and mitosis. Have a check out of my meiosis video to make sure that you don't confuse the 2. Let's just remember that in meiosis, the number of chromosomes split down in half. They're haploid cells in gametes, which are sperm and eggs, but in mitosis, it's always in the diploid form. It's always the right number of chromosomes. That's 46 in humans, arranged in 23 pairs. And are you interested in more of our biological resources? Well, why not check out more of our videos at the CyberTeachers channel? And if you found this CyberTeachers video useful and interesting, why not like and subscribe, and we'll keep the CyberTeachers content coming.